Hello everyone, welcome back to the session. So in today's class, we are going to discuss about the topic cell cycle. See, we are in the end of the cell biology chapter because we have started from the cell theory and we have continued with the plasma membrane, nucleus and all the most important cellular organelles. Now in today's class, we are going to discuss about the cell cycle. Okay, now in cell cycle and after finishing this we are going to discuss about the cell division. After finishing the cell cycle and cell division, almost the all topics which is coming under the cell biology has been completed, and your duty is after watching the class, whatever the book you are having, just go through it and try to solve more and more questions in each topic. This is how you have to be prepared. And in today's class we are going to discuss about the cell cycle and the first introduction of the cell cycle. How can we define the cell cycle and what is exactly done under the cell cycle? See, the cell cycle is a highly regulated sequential series of events by which a wall grows, duplicates its genetic material, divides to form two genetically identical daughter cells. So you should know that one cell is going to grow, and in which within the cell their genetic material has been duplicated. One genetic material has been duplicated into its copy. Now after that, after the cell has reached certain size, it tend to divide. Okay, so divides to form two genetically identical daughter cells. Finally one cell divides to two different cells. So this is what cell division. And the cell cycle is, so before the cell division is going to happen, the cell cycle has to be completed, and this cell cycle is not a random phenomena, but it is a highly regulated cycle, and it is a sequential events, one after the another. Many events is going to happen in the cell cycle. Okay, so series of events. So and what are all the series of events? First the cell has to grow, and after that it has to duplicate its genetic material, and finally one cell has to divide to two daughter, and the most important thing will be the genetically identical daughter cells. Okay, so this is what exactly happened in the cell cycle. Now the term cell cycle was proposed by Howard and Pelk. So they were the scientists who proposed the term cell cycle using the autoradiographic studies, and they also didn't randomly name the term cell cycle, but before they are naming this word cell cycle, they are using certain studies, more specifically the autoradiographic studies. Now, so this is what exactly the cell cycle. In cell cycle, these are all the phases we are going to study. So what in each phase, what are all the events are going to happen? We are going to discuss about that. See, we are having G1 phase, S phase, G2 phase and M phase. Okay, and between this M phase and G1 phase there is a phase which is called as a G0 phase, and we are going to study about the importance of G0 phase also. Just listen to me here. See, this G1 phase, this phase is considered as a cell growth phase, right? See, this G1 phase is considered as a cell growth phase, which means all the cells are in a growing stage, and that stage is called as a G1 phase. And also this G1 phase is considered as a preparatory phase for the S phase. This is very very important, this G1 phase where all the cells are going to grow, and also the G1 phase is the preparatory phase for the S phase. Now, so this S phase, what is exactly happening in case of S phase, and this is very very important, where our genetic material, that is the DNA, is going to replicate. In S phase, the DNA is going to replicate. So DNA replication or the synthesis of DNA has been taking place in the S phase. So that is why it is called as a synthetic phase. Right now, G2 phase, and G2 phase we are calling them as a preparatory phase for the M phase, and M stands for the mitosis, and that is why we are saying this G2 phase as a preparatory phase for the M phase. So like in the same way as G1 is considered as a preparatory phase for the S phase, and G2 phase is considered as a preparatory phase for the M phase. Now what is exactly happening in this mitotic phase? That is the M phase. So here see, we are having different phase under this mitotic phase. The first one will be the prophase, metaphase, anaphase and telophase, and followed by cytokinesis, and all these process we are calling them as a cell division. See here, the cell division which is exactly taking place in the M phase, and you should know what is the difference between the cell cycle and the cell division. Cell cycle in the sense, cell cycle means the G1, S phase, G2 phase, M phase and G0 phase. These are all the phases which is called, we are calling them as a cell cycle. And the cell division means this M phase, the phases which is coming under this M phase is very important to divide the cell. So that is why we are saying the cell division is taking place in the M phase. This can be expected in the question. Now what about this G0 phase? G0 phase, it is also called as a resting phase. It is also called as a resting phase, and also it is also called as a quiescent stage. Quiescent stage or resting stage or G0 stage, right? See, the word G refers to gap. Gap 1, S phase, G2 phase, M phase and G0 phase. And one more important point will be here. See, this G1 phase, S phase, G2 phase, all these phases we are calling them as an interphase. So you should know, whenever in your exam paper you find the word interphase, and that interphase includes G1 phase, S phase and G2 phase. Okay, so this will be the overview of this cell cycle. Now we'll dive deep into it. See, the overall phases of the cell cycle, just now I have explained you with a picture. Just imagine that the cell cycle has been divided into two major phases, and that will be interphase and M phase. Just now we have studied this interphase contains the G1 phase and G0 phase, S phase, G2 phase, G1 phase, G2 phase and S phase. All these phases together we are calling them as an interphase, and that interphase constitutes approximately 90 to 95% of the cell cycle, and remaining 10 to 5% of the cell cycle will be the M phase, that is the mitotic phase, isn't it? See here, the interphase is the longest and most metabolically active phase. This phase is called as an interphase, right? So this is the longest and most metabolically active phase. Now what comes to the G0 phase? See, here the cells are alive, but it is metabolically active, but it is not undergoing any growth, any cell division process. The cells are in a rest stage. So that is why we are saying this G0 phase. So as the cells are not doing anything, you should not know the cells which is entering into the G0 phase, which is a dead cells. No, it is not. Okay, the G0 phase, the cells which is entering into the G0 phase, it is not going to divide. It is not going to divide, but it is metabolically active cells. So this is the overall phases of the cell cycle. And the interphase. See here, just now I have explained you, the interphase consists of three sequential subphases, and the first phase will be the G1 phase, and the second phase will be the S phase, and the third phase will be the G2 phase. These three phases we are calling them as the interphase. So the first phase will be the G1 phase, and the G1 phase we can call them as a first gap phase. So in this gap phase, major events is going to happen, which means what are all the things which is required for the cell synthesis, the DNA synthesis, which means the replication of DNA which is taking place in the S phase. And what do we have studied about this G1 phase? The preparatory phase for the S phase is called as a G1 phase, and you should know what are all the things which is required for the DNA replication. All these process will be happening in the G1 phase. Now major events is going to take place in the G1 phase, and the cell reaching certain size. Only when the cell is attaining certain growth, it is tend to divide. isn't it? So that is why the cell attain a proper growth only in the first gap phase, that is the G1 phase. And the synthesis of RNA and proteins. See, the synthesis of RNA and proteins is very important in the DNA replication. In the DNA replication, the most important enzyme will be the DNA polymerase, and that is nothing but a protein. And all these things has been synthesis in the G1 phase, but it is inactive. But it is inactive in condition. When only whenever it is entering into the S phase, it tend to become active, because only in the S phase the DNA polymerase activity is required, not in the G1 phase. Now you should know what are all the things which are required for the DNA duplication, that is exactly prepared in the G1 phase, but all these things will be in an inactive manner. Only when all these complexes is entering into the S phase, it becomes active, and that has been activated by the complex which is present in the S phase. I repeat, what are all the things, what are all the materials which are required for the DNA replication has been prepared in the G1 phase. That is why we are calling this G1 phase as a preparatory phase for the S phase. And you should know what are all the materials which are prepared in the G1 phase, that will be in an inactive manner, and only when it is entering into this S phase, then only then it will become active. Why? Because the complex which is present in the S phase is going to activate the materials which are entering from the G1 to S, because their importance is needed in case of S phase, not in the G1 phase. This is very important. Now, increase in the cytoplasmic volume. See, only when the volume of cytoplasm is increases, there will be a sufficient volume even after it has been divided into two cells. That is why increase in cytoplasmic volume is very important. See here, this is a cell which is contain 50% of the cytoplasmic volume, just imagine, and this is a cell which is having the 100% of the cytoplasmic volume. Now this is reaching the proper size, and this 50% is not attaining the proper size. Now if this cell is undergoing a cell division, here 25 and here 25, and there will be no sufficient division, there will be no sufficient materials after the cell division, and the divided cell which is not in use. Okay, only after the cell has been reaching the 100% cytoplasmic volume, after then it will be divided into two cells, then it will be sufficient for their survival. So this is why it is very important to cell to reach certain size, and the cytoplasmic volume has to be increased. So all these things are very important. Now, organelle duplication. See, this is very important. You should have to know that only the DNA duplication has been taking place in the S phase, but not the duplication of the organelles. Now here in G1 phase, the organelle duplication, that is like mitochondria, endoplasmic reticulum, all the important organelles has been duplicated only in the G1 phase, but not in the S phase. So this is very important. Just see here, what will be the very important important material which is present in the G1 phase. Presence of restriction point, that is the R point. Only when the cell has reached a certain size, this cell is allowed to enter into the S phase. If not, it has been restricted within the G1 phase, it undergoes a modification, and only after they reach a certain size, it is allowed to enter into the S phase. So that restriction point is called as the R point. Okay, so that R point is restriction point. So that restriction point is present in the G1 S phase gateway. Right, now see here, this R point, the cell, see, this is the R point, and that point is very important, whether the cell from the G1 phase is okay to enter into the S phase or not. If the cell is reaching certain size and all the conditions of the cells in proper, only then this R point is allowing the cell from the G1 phase to the S phase. If not, no cell has been entered from the G1 phase from G1 to S phase. So that point is called as a restriction point or the R point, right? If not, the cell is attaining a proper size, that all those cells has been entered into the G0 phase. right? So this is what the importance of the G0 phase. Now what is the biological significance of this G1 phase? Cells lacking growth factor stops at G1. See, what do we have discussed before? The cell has to reach certain size in the G1 phase itself, and if not, for to attain a proper growth, a growth factor is important. If the growth factor in a particular cell is not that much efficient, the cell will not reach a proper size and it will not be enter into the S phase. It has been restricted by the restriction point and the G1 phase, isn't it? So that is why all those cells has been enter into the G0 phase. Now most cancer causing mutation affect this phase. Very, very important in an exam point of view. See here, so this is what a cell. So this is a cell, and if this is present in the G1 phase and this is in the S phase. Now the cancer cells will not reach a proper size. All those cells which is a rapid repeated uncontrollable cell division is nothing but a cancer, and in those cells not all these cells are in a proper size, and all those cells has been restricted in the R point itself. So that is why cancer causing mutations affect this phase, right? So this is very important. Now coming to the S phase. So this S phase is very important, though S phase is also called as the synthesis phase. So here, what are all the major events which is taking place in the S phase? That is the DNA replication, histone protein synthesis. See, the histone has to be wrapping up the, histone is a protein, it is a positive charged protein, and the DNA is a negative charged nucleic acid because of the presence of the phosphate group. Now, so this histone proteins wrapping up the DNA. For the packing of the DNA, this histone protein is very important, and the histone protein is also synthesizing in an S phase. This is the major event. Now, the formation of sister chromatids in the sense the chromosome. Okay, so this is a chromosome. One chromosome have two sister chromatids. Now all these sister chromatids is also formed in the S phase. Now centrosome duplication. The centrosome means, so the point which is connecting the two chromatids, this is the center point, and this center point is called as a centrosome, and the centrosome is also getting duplicated in the S phase. So see here, not only the DNA synthesis and the DNA replication is taking place in the S phase, but also histone protein synthesis, formation of sister chromatids, and centrosome duplication. All these processes will be taking place in the ES phase. Now DNA content doubles. This is exactly the exam point of view. So this is very important. From these lines we can expect our question. See here, initially the content of the DNA will be C. This C refers to the amount of DNA. Right, so the C refers to the amount of DNA. See, what do we have discussed here? The cell is getting duplicated in the S phase, and the cell is reaching certain growth in the G1 phase. Now the amount of DNA in a cell which is present, the amount of DNA in a cell which is present in the G1 phase, it is 2C. The amount of DNA which now the 2C containing cell, it is going to entering into the S phase, and in S phase this DNA is getting duplicated. Now the amount of DNA will be converting into 2C to the 4C. So here is where the amount of DNA is getting doubled. Just remember here the cell content, DNA content is one double, but the chromosome number remains unchanged. So here where we can expect a question. Okay, see here, so this is what a cell, and in this cell we are having 46 chromosome. So just to listen, only the DNA content, that is the amount of DNA from 2C has been doubled into the 4C. The chromosome number, but the chromosome number, what is the chromosome number seen in the G1 phase? That will be the chromosome number in the S phase also. Now see here, the replication is semi-conservative process. We know that replication is semi-conservative, and I'll explain this process very detailed manner whenever we are discussing the molecular biology. Just know as of now, know that replication is semi-conservative, and the word semi-conservative means, so whatever be the generation be, so this is what a parent DNA, and this parent DNA, just look at this. So this is a parent DNA molecule, and this has been divided into a single stranded with the help of an enzyme called as a DNA helicase. So all these processes will be discussed detailedly in the molecular biology chapter. Just know why I am saying this replication is semi-conservative process. So this is what a parent strand, and this parent stranded has been divided into DNA is originally double-stranded DNA, and this double-stranded DNA with the help of an enzyme it has been divided into single-stranded DNA, and from this single strand with the help of another important enzyme called the DNA polymerase, a complementary strand is going to synthesize. Now we are having one parent DNA, another newly formed DNA, and also here one parent DNA and another newly formed DNA. Now again this DNA, double stranded DNA is divided into a single strand. Again this will be divided into a single strand. Now here also we are having a parent strand. So whatever be the generation be, our parent strand will continue to exist. So that is why we are saying this replication process is a semi-conservative process. It is not completely conservative, but it is semi-conservative. So each chromatids contains one parental and one new strand. So that is why we are saying this replication is a semi-conservative process. So this is an important thing with respect to the synthesis phase, that is the S phase. Now comes to the G2 phase, and this G2 phase is also called as a second gap phase. And what are all the major events which is taking place in the G2 phase? See, the first important event will be the synthesis of tubulin proteins. And what do we have discussed with respect to this G2 phase? And this G2 phase will be the preparatory phase for the M phase, and we are saying that in the mitotic phase, that is in the M phase, most of the cell division is going to taking place. Now what are all the materials required for the cell division? All those materials has been synthesized in the G2 phase itself. So the synthesis of tubulin proteins is very, very important, because tubulin proteins is important. That is what we are saying, that as a microtubules. Just remember what are all the things we have discussed whenever we are dealing with the microtubules. Now this tubulin proteins together make up the microtubule, and microtubule is mandatory for the synthesis of the spindle apparatus preparation. Now DNA damage repair. Now the DNA has been duplicated in the S phase itself. Now whenever the DNA duplication has been occur in the S phase, it has been enter into the G2 phase, and you should know if any DNA is getting damaged and the repair of if there is a DNA repair damage has been taking place in the G2 phase itself. Final growth before division. So final cell growth. So if any cell it is not attaining further in a growth, if further any process has to be added on, all those growth final cell growth has been taking place in the G2 phase itself. Only then it has been entered into the cell division phase, that is nothing but an M phase. So this is very important. Now in the G2 phase, the cell has been prepared for entering into the mitosis phase. Now what is with respect to this G0 phase? This G0 phase is also called as a quiescence phase. G0 phase is very important for G0 phase is also called as the quiescence stage. Just see here, the cell exit from the cell cycle temporarily or permanently. Say for an example, let me explain you. Just now we have studied the G1 phase is a preparatory phase for the S phase, and in the G1 phase the cell has to attain particular growth. If the growth factors in a cell is not proper, if not that much efficient, and that cell has been exit from the cell cycle, which means it is not going to continue with the rest of the cell cycle. Now that is why they are exit from the cell cycle and it is entering into the G0 phase. Either it will be temporary or it will be permanent. So this is important with respect to the G0 phase. Cell remains metabolically active but do not divide, and this is an important statement. This can be expected in the exam. Cell remains metabolically active but not undergoing a division. So you should not think that whenever the cell is entering into the G0 phase, you should, as a cell is not doing any function, you should not think that cell is dead. That cell is alive, it is metabolically active, but only thing, when compared to the other cell, this cell is not entering into the cell division. This cell do not divide. So these are all the important points under the G naught phase. Very important. As we have mentioned before, as we have studied here, the cells will exit the cell cycle. Either it will be temporary or it will be permanent. If it is a permanent G naught phase, the neuron cells, cardiac muscle cells, all these cells are entering into the G naught phase and it will not enter into the cell cycle further. Temporary G naught phase means the liver cells and the lymphocytes, temporarily it is entering into the G naught phase, but again it will be enter into the cell cycle and continuing its division. That is why whenever any damage occur in the brain and in the heart, it is very difficult for doctors to recover a patient. Why? Because the cells which is in a damaged condition, they will be entering into the G naught phase. That will be not reverse back. It is a permanent G naught phase. Right, so this is very important, and the cells in G naught can re-enter G1 under favorable condition except permanent G naught cells. So you should know the neurons and the cardiac muscles, that will be, those are all the cells which is entering into the G naught phase, which is not undergoing any of the cell division. So this is very important. That is why if any damage occurring in the brain cell and the heart cell, it is very hard to recover a patient, right? So this is very important. Just see here, so this is the cell cycle phases, and the G1 phase, M phase and G2 phase, and G1 phase, S phase, G2 phase and M phase, and G1, S and G2. Together we are calling them as an interphase. 90 to 95% of the cell cycle will be this interphase. Remaining 10 to 5% will be the coming under the M phase. And G naught phase, just now we have studied what are all the specifications of each and every cycle. See here, look at this picture. See, G1 phase, the cell will grow and perform a normal function. This is undergoing a cell growth. So the cell growth phase is called as a G1 phase. Now after the cell is attaining certain growth, it will be entering into the S phase, where the DNA replication of that particular cell occurs. Now followed by the G2 phase. In the G2 phase, now the cell is getting ready to undergoing a cell division, and to enter into the mitotic phases, that is the cell division phase, certain materials are mandatory, and all those materials has been synthesized in the G2 phase. Now the cell is ready for the mitosis. It will be entering into prophase, metaphase, anaphase, telophase, cytokinesis, and all these cell division will be discussed detailedly tomorrow. As of now, we are going to discuss only the cell cycle. Now M phase. So in M phase, what is exactly happening in the M phase? More specifically, karyokinesis and the cytokinesis. Always the word kinesis refers to division. Kinesis refers to division. Karyo means nucleus. The word karyon refers to nucleus, and the word cyto refers to cytoplasm. So the division of nucleus is karyokinesis and the division of cytoplasm is cytokinesis. So this is what we are calling them as the M phase. Now see here, and in karyokinesis, many important things will be taking place. So 4 stages will be coming under the process karyokinesis. That is the prophase, metaphase, anaphase, telophase and cytokinesis. All these things we have discussed these things in the cell division chapter, but as of now we are going to discuss about only the cell cycle. Just to know that what are all the checkpoints which is present in the cell cycle. What are all the checkpoints? Checkpoints are very, very important. See here, as we have mentioned before, the G1 phase, the cells from the G1 phase has to enter into a S phase, and from the S phase it has to enter into the G2 phase, and from the G2 phase it has to enter into the M phase, and this is the one complete cell cycle. Now the cell, in all these areas certain checkpoints will be present, which means this is like a security guard. The cell which is coming from the G1 phase to the S phase has been checked by this G1 checkpoint. So in this G1 checkpoint, what are all the things which are exactly happened, and in the S checkpoint what is exactly happened, and in this checkpoint and in this checkpoint what are all the events going to happen. So this is what we are going to discuss now. Just look at this. accurate DNA replication. So these checkpoints are repeatedly checking whether the DNA replication is accurate, 100% accurate or not, whether the DNA replication is proper or not, and whether all the chromosomes has been segregated properly or not. Chromosomal segregation process, it is a part of the cell division. We are going to discuss about that, right? Just know that. So these are all the things which are ensured by the checkpoints. Now coming to the G1 checkpoint. In this G1 checkpoint, this G1 checkpoint is checking whether the cell has reached a certain size, because these are all the cells which is entering from the cell G1 phase to the S phase. Now in these checkpoints, they are checking whether the cell is reaching a proper size or not, whether the nutrient availability of these cells are proper or not. If there will be no proper nutrition, there will be no proper cell division. So that is why they are ensuring that whether the nutrient availability is proper in that particular cell or not. Now the DNA damage, whether any DNA damage, so before the DNA has to be enter into the replication process, they have to know, this checkpoint have to know that whether the DNA already present in that cell is in a proper condition or not. So all these things which is checked by this G1 checkpoint, only then it will enter from the G1 phase to the S phase. Now this G2 checkpoint, they check the completion of DNA replication. Now just listen to me. G1 phase to S phase, and S phase to G2 phase, G2 phase to M phase, and M phase to G1 phase. And in the G2 phase, they are in the G2 checkpoint, they are checking whether the DNA has been replicated properly or not, and whether the DNA replication is properly completed or not. And if there are any damage, DNA damage, we are going to repair the DNA damage, and only after repairing the damage, it has been entering into the M phase. So all these things has been checked by this G2 checkpoint. Now M checkpoint, this M checkpoint is also called as a spindle checkpoint. Now see here, the spindle checkpoint, it is checking the proper attachment of chromosomes to the spindle fibers, whether the chromosome has been properly attaching, the spindle fibers is properly attaching to the chromosomes or not. Okay, only after the spindle fibers has been attached to the chromosome properly, the chromosome has been segregated in a proper manner. So this is why we are saying to ensure the chromosomal segregation has to be proper, 100% accurate. The proper attachment of chromosome to spindle fibers is very important, and whether the attachment of the chromosome to the spindle fiber is proper or not, that has been ensured by this M checkpoint or the spindle checkpoint. So if there is any failure in case of spindle fibers attachment to the chromosome, it results in chromosomal abnormalities. Now, what are all the molecular regulations of the cell cycle? How the cell cycle is getting regulated at a molecular level, and what are all those? And what are all the proteins which are involving in regulating the cell cycle? And the first important thing will be the cyclin. Cyclins are the regulatory proteins. Why they are called as the regulatory proteins? Because they are involving in the regulation of the cell cycle. That is why we are saying the cyclins as a regulatory proteins. Levels rise and fall cyclically, and very important thing will be this point. The cyclins will be, some of the cyclins will be present only in the G1 phase, and it will be absent in the S phase. It will be present in the G2, and it will also present in the M phase. So you should not think the cyclins will all be present in an equal amount in all the phases. That is exactly wrong statement. The cyclins level will be rise in certain phase, and it will be fall in certain phase based on their requirement. And see here, cyclin dependent kinases, this is very important. So if cyclins are very important for for the activation of the cyclin dependent kinases. So that is why the name cyclin dependent kinases. So as the word kinase, again it is an enzyme, and their level remains constant. Okay, see here, so what do we have? G1 phase, S phase, G2 phase, and M phase. These are all important phases. The CDKs, that is the cyclin dependent kinases, will be present in all these things, and the cyclin dependent kinases will be in an inactivate manner. Right, now The cyclin dependent kinases is getting activated only with the presence of this cyclin, that is a regulatory proteins, and this cyclins will not be present in all the phases equally. It will rise in some phase, it will fall in some phase based on the requirement. Cyclin plus CDK complex drives all the cell cycle transition. So we have studied cell growth will be taking place in G1 phase, cell duplication will be taking place in the S phase, preparatory for the M phase will be the G2 phase, and all these phase, all the materials has been properly synthesized with the help of this cyclin and CDK. So in one line we can say that only the cyclin and the cyclin dependent kinases drives the complete process of cell cycle. Okay. Now, this is very important slide you have to keep in mind. In G1 phase, cyclin D will be present and the cyclin dependent kinase 4 and 6 become activated. In G1 phase and G2 phase in the checkpoint, cyclin E will be present and cyclin dependent kinase 2 will be present. And in the S phase, cyclin A will be present and cyclin dependent kinase 2 will be present. And cyclin 2, CDK2 will be activated by this cyclin A. And in the G2 to M phase, cyclin dependent kinase 1 will be activated by the cyclin B. This can be expected in a question, very important. Kindly make note of it. Now, MPF. MPF is very important, a checkpoint, that is maturation promoting factor. Mitosis promoting factor, or we can also say that as a maturation promoting factor. And this MPF, what do we call this MPF? Composed of cyclin B and CDK1. Cyclin dependent kinase 1. Just look at this picture. Cyclin B and CDK1, it is very important for mitosis promoting factor. So this factor is very important in the mitosis phase. So that is why mitosis promoting factor. So the cyclin B and CDK1 come into picture. And this triggers this complex MPF factor is the one responsible for triggering the cell to enter into the mitosis. What are all the functions of this MPF factor? The nuclear membrane breakdown. The nuclear membrane breakdown will be considered as a prophase, and in this prophase, this MPF factor is getting activated and it is involving in breaking the nuclear membrane. And chromosomal condensation of chromosome will be taking place. Spindle fiber formation, and see, just don't confuse with this. All these important terms have been discussed in the cell division in the tomorrow's class. As of now, know all these factors are important for the cell cycle and what is its function. And the spindle fiber formation will also be taking place by this MPF. See, this is an important keyword in an exam point of view. Just look at here. This MPF activity peaks, which means the MPF activity will activate in a higher level during the metaphase and declines after the cyclin degradation. When the cyclin B is getting activated in the metaphase, at the end of the prophase and during the metaphase, and if the cyclin B is getting degraded, automatically this MPF activity is also getting down. Okay, so this is very important. Now, see so we will discuss this cell cycle inhibitors for this P53 and the P21 in tomorrow's class. Just tell me, we are going to discuss about this questions. So see here, we have discussed in today's class what is cell cycle, and in each cycle what are all the things are going to happen, and the leftover part will be the cell division, and the P53 and P21. What will be the importance of all those? We are going to discuss about that tomorrow. So first question will be, which of the following statements regarding the S phase of the cell cycle are correct? So in the S phase, they are going to do the DNA content is double and chromosomal number doubles, and this will be the wrong statement, so we can eliminate the first and second option. And so the first A and C will be eliminated from the option. Histone synthesis occurs. Of course, the histone synthesis will be there, isn't it? Now the centrosome duplication occurs, so the answer will be option B. And we'll discuss this question in tomorrow's class. See here, which protein complex directly triggers nuclear envelope breakdown? So the nuclear envelope breakdown will be done by the MPF factor, and the MPF factor consists of the cyclin D and CDK1. And see here, this is very important. The diploid somatic cell in G2 phase has diploid somatic cell in G2 phase has chromosome number, and what will be the DNA content? Definitely the DNA content will be 4C, and what will be the chromosome number? That will be the 2N. It is diploid and it will be 4C. So this is very important. See, just look at me. In G1 phase, just make a note of it. In G1 phase, the chromosome number will be 2N, and the chromosome content will be 2C. And in the S phase and the G2 phase, the chromosome number will be 2N, and the chromosome content, DNA content, will be 4C. Kindly make a note of it. Which event occurs only once per cycle? Spindle formation, DNA replication, chromosome condensation, nuclear membrane breakdown. And when compared to all these events, the DNA replication is occurs only once per cycle, that is in the S phase. Just see here, G1 phase we are having a restriction point, in S phase we are having a histone synthesis, and G2 phase DNA repair is taking place. So the answer will be option 231, that is option A. Now, anaphase and everything will be discussed tomorrow. And which of the following is not a function of the MPF? Mitosis promoting factor, chromosomal condensation. Chromosome condensation is a part of the MPF. Nuclear membrane breakdown is a part of the MPF. And DNA replication in spindle fibers assembly, again it is a part of the MPF. The DNA replication will be taking place in the S phase, not in the M phase. And MPF will be the part of the M phase. And this will be discussed in tomorrow's class. And a cell permanently arrested in the G0 phase is characterized by, a cell permanently arrested in G0 phase is characterized by absence of metabolic activity. No, it is wrong. It is metabolically active. Inability to synthesize RNA. We'll see loss of division potential. So this will be the proper answer for this question. Incomplete DNA replication, inability to synthesize RNA. So they were metabolically active, but the only thing will be they are not involving in any of the division process. Okay, so in today's class we have completely discussed about the cell cycle, and only leftover part will be the mitosis phase. We are going to discuss all the phases one by one, and after that we are going to discuss about this P53 and P21. What is meant by P53 and 21? Okay, so thank you so much. We'll meet you in tomorrow's class.